start date.
It's not going to This gives me so much anxiety, but I understand. Yeah.
I have email now. Share.
Oh, you have it?
Right now. It's very long.
Where's Steve Nelson?
Sure.
Have you tried the missing voice?
Has another or is it done?
Submitted as of yesterday. I got a notice from the NIH. He said your grant status has been. I was
Did
like,
you need to submit this as a
yeah, because I have other stuff to do, so I'm like, I just need this to be checked off. I'm like, that's
It's on.
Oh, there it is. It's coming.
yeah,
And then the microphone, you can just turn on whatever.
super submitted.
submitted to submitted?
Yeah, I know, what, okay.
You really submitted this one?
You know that.
There's water?
Yeah, I think you can turn the light like this.
Oh, off like this?
No, not like this, but what is the previous
When it comes back, then it's.
Like turn it on?
That's what-
Yeah, because I will be introduced, but then maybe.
Oh, and then after. Okay, yeah.
What are you doing?
I usually have to do this in my privacy.
Good, review for lunch. Lunch also.
Yep.
Should be.
Yeah.
It's more people are slow down. How long is the
Yesterday, they had 185, but refresh. Use the probably 6 times. But it was still 2 days.
You're checking the CBS school as well. I always emotionally prepare myself. By myself. Just take what I'm gonna get.
With a strong drink. Something to numb the pain.
Stay away from the lab.
This is that we usually use.
It should work. Is it working? No?
No, it doesn't.
Okay. You probably just do that. Here's a backup. Is this
I'd like to do the
USB-C?
or the DAB application, so you can, there's a lot of sense on it.
That was good. Yeah, didn't connect with Anna. I'm not sure to see her yeah, it didn't. It's eventually used Ryan's connection, but I didn't realize that she's a different.
Yeah, she's a different.
No, you're good.
She did.
No, she was with
Oh, that's
Chris.
right. She was a president.
Yeah.
Sorry. Oh, right.
Yeah, there's another project I have to talk to her about this.
I'm sorry. What was it?
No, the Zoom link needs to be screwed up as a postdoc. Just like sitting there. So, anyways, we just figured out. I wasn't sure if she was there or not.
Oh, she was.
I wrote her. So,
Because it was just kind of weird.
yeah, just.
You met Susie.
Oh, no. Yeah, it was really intentional stuff. So it's real life sequence.
All right. Sabina.
Yep.
So, I will start. Right?
Yep.
All right. Welcome everybody to the GSI seminar. Today we have a guest, Eric Lai, who's a member of Memorial Sloan Kettering Cancer Center in Developmental Biology program. He's a professor in Weill Cornell Graduate School of Medical Sciences. And Eric got his undergraduate degree in biochemistry from Harvard College. And something that I didn't know, that he worked with Gary Rufkin actually during his undergrad on homeobox gene in C. elegans. And after his bachelor's degree, he moved to California, and he did his PhD in University of California, San Diego with James Mazzaconi. There he studied Drosophila neurodevelopment, and importantly, he identified negative regulatory elements in the boxes in 3' UTRs of several genes, which was very important finding that later led him to realize that when microRNAs were discovered, that those were targets for microRNA regulation. So it was a very important insight and led, of course, to him getting more interested in microRNAs. So which he did when he was a postdoc at the University of California, Berkeley, with Gerald Rubin, where he studied notch signaling and also microRNA research. He started microRNA research, which he then continued in his lab in Sloan Kettering, where really his career flourished. And it's I know that Eric really has exceptional productivity in a wide range of topics related to small RNAs, but not only small RNAs. But in the field of small RNAs, he contributed to, he's one of the leaders in the field, and he contributed to understanding of microRNAs, his famous discovery of mirtrons, which are alternatively biogenesis, which is different from canonical microRNAs. His lab was first to identify siRNAs that are not microRNA in flies, coming from hairpins. I especially like his work on recent work on control of selfish meiotic drive by RNAi pathway, the canonical RNAi pathway. He studied piRNAs, a lot of work on small RNA evolution and genomics, and he didn't forget neurodevelopment. That was his topic during his training, and he also continues to produce interesting work about neurodevelopment regulation in mice as well as Drosophila. He's a very collaborative person, and people in the audience, Daniel and Nelson, have been his collaborators, and he's also president-elect of North American Drosophila Board, Fly Board, congrats. And we will hear more about Eric's recent. Yeah, I forgot to tell you that he's a great guitar player, and he has songs on the internet that you can find related to science, actually, and microRNAs. So, yeah, multi-talented.
Oh, yeah, it's on.
Okay, well, thank you, Ella, for the kind invitation, and it's really wonderful to be here. We're meet some new people, but also have a lot of friends here, so it's great to spend some time here at BU. So I'd like to begin by sort of remarking the past few years have been quite extraordinary for good and bad ways, but certainly truly out of the ordinary. But sort of one interesting byproduct is the fact that people outside of science are aware of what RNA is and know what RT-PCR tests are and mRNA vaccines, for better or worse. There are people who don't support these, but it's been kind of an interesting time to be able to have outreach to 9 scientists. And I think it's very important as scientists that we take efforts. We see in the coming election how much anti-science rhetoric there is. We do need to reach out to the public at large about what we do and why it's important. And the spotlight now for a lot of stuff is on RNA, which is a great time to be able to relate some of the things we do in RNA processing and regulation and their impacts on biology. So We study a number of different paradigms that have to do with the generation of alternative isoforms by alternative splicing or alternative 3' UTRs. But today I'm going to focus on some of the things we do. As I'll mention, I'm a long-standing interest in microRNAs and RNA interference, and I'll tell you two stories that have to do with sort of one of each of these things. There'll be sort of two very different half talks, one dealing with biochemical mechanisms and the other one about biology. So to introduce, and we have some of the founders of the field here in the audience in the department. You know that from Andy Farr and Craig Mella's work 25 years ago, we were introduced to this magical property of double-stranded RNA to be able to silence homologous transcripts in worms, and all that had done some of the genetics of RNAi very early on. And it turns out that this silencing process implied something which is degrading the target transcripts that are complementary to the double-strand RNA, and that turned out to involve a short RNA that is associated with a special argonaut, which has a nuclease and endonuclease activity, and in human cells this is ACO2. So some of the basic properties of this is that, as first conceived in worms and certain other invertebrates, you can use a long double-strand RNA trigger, which is complementarity to the target gene you're trying to silence. But the double-strand RNA precursor is not the thing that's doing the silencing. That has to be metabolized into about 21 nucleotide RNAs that can fit inside of the argonaut protein. And those are the things which guide this nuclease to downregulate targets. Now, also during the 90s, there were parallel studies also in worms from Gary Ruvkin and Victor Ambrose, which showed that worm development was highly dependent on another type of double-stranded RNA, but in this case a hairpin double-strand RNA that produces a small RNA about the same size as the effectors in RNAi. But unlike these, they have only partial complementarity to target genes. And this is the famous LID4 RNA, which is produced from this hairpin here. That short RNA regulates a very critical developmental timer that controls early larval development in worms by binding to complementary sites in the 3' UTR of this gene Lin14 to suppress it. And a very exciting time for our field because although we worked with Craig, Andy and Craig were recognized with the Nobel in 2006, and just a couple weeks ago, Victor and Gary received this year's Nobel Prize. So going beyond worms, we now appreciate that these really are manifestations of quite ancient regulatory strategies for homology-based silencing using short RNA molecules. Plants are not recognized in these prizes, but in fact, plant RNAi was worked out its biology. It's the small RNAs that occur in the plant RNA pathway were recognized before the manifestations of RNAi in animals. So we have plants and animals. We even have the same systems working in archaebacteria. So these are very ancient regulatory systems that are used using this foundation of the template of the pathway that makes the small RNA and then mediates and identifies and mediates target gene silencing. This is used in many different ways in many different organisms. So many of us in the field have been exploring not only the canonical model organisms, but how you can use this not only to manipulate, but also find new kinds of pathways across different organisms. That's a little bit what I'll talk about today. Because it's kind of a special time, I did want to highlight that in 1992, I was an undergraduate and I did my thesis in Gary Ruvkin's lab. I worked on homeobox gene project, but that was the time when Lin14 was being figured out, and a very formative time. It sort of taught me everything I know about development and genetics, and also gene regulation by small RNAs and transcription factors, pretty much everything I've studied the last 32 years. And a wonderful time with Gary yesterday. Okay, so as I said, I'm going to break this talk up into sort of two sub-talks, and they really kind of hit the way I approach many different projects we study. one, we want to know mechanisms of how gene regulation work. How do these processes function? And then we want to know what's the biological meaning of this, and preferably in intact organisms, not just in cell culture. So we want to know biology. Why do we have gene regulatory strategies? We want to find mutant phenotypes when we break those regulatory strategies. So how does things work, and then what are they good for? The first half I will spend talking about a new mechanism we've identified, which restricts your ability to do microRNA silencing, microRNA-mediated silencing. And the upshot of that is that when you remove that break, you can do better silencing. Pretty interesting. So we think that there's some interesting implications of how we might be able to use that knowledge therapeutically in the future, perhaps. So that's all going to be in mammalian cells and in mice. Second part, I want to talk about RNA interference. So not microRNAs, but actually RNAi. What is that good for? We know it's very good for doing knockdown experiments, but somehow less appreciated what is the normal biology of RNAi. So we found that RNAi is super critical. Maybe one of its most important phenotypically overt settings of RNAi is to suppress parts of the genome that are warring against each other, so-called intragenomic conflicts. And a very special kind of conflict that occurs between the sex chromosomes. So that'll be the second part of the talk. This will be in flies. Okay, so the first part is about this break on small RNA pathways in mammalian cells. This is the core pathway that became outlined quite early when I started my lab in 2005. You start with a transcript which has one or more inverted repeats of a certain size that are identified as substrates for a nuclear RNase-3 containing machinery, where Drosha is the enzyme that cuts out the hairpin. And then the cytoplasm, it's cut again by another RNase-3 enzyme called dicer to make a small RNA duplex. And these things then associate into an organoid protein and mediate, and try to find those targets that are special targets for gene regulation. And shout out to Nelson, because he identified in the first paper, the first of 3, on the large-scale identification of microRNAs, once we knew that they have certain biochemical features that you can take advantage of for cloning. Now, it turns out that I draw this as an arrow, arrow, arrow to here. You get the sense that this is a straight shot down to make new microRNAs. That's definitely not the case, but it's just simplest to write it that way. So there are a lot of regulatory steps. Every pathway has to have regulation, whether you know about it or not. Because in biology, we're always optimizing, we're always trying to have the ability to do things a little bit different, adjust to condition or cell types, what have you. The other thing that it turns out is that there are many additional ways to get small RNAs that eventually get into an organoid protein. And so all these non-canonical pathways, by one means or another, bypass either Drosha or dicer. There's a whole set of pathways that we've studied that bypass Drosha. As Daniel knows, that there's also a special pathway that bypasses dicer. We've also studied a so-called dicer-independent pathway. There are still additional pathways that we are investigating at the moment for non-canonical and or regulated biogenesis. So the picture kind of keeps getting more and more complicated the more you spend time with it. But that'll be for another time. Today I want to talk about the canonical pathway. So just go back to the simplified picture. So again, we have these two RNase-3 mediated cuts of the primary to precursor microRNA to give you this RNA duplex. Then you have to get that into an organoid protein. And for the first half of this talk, we're going to start with the microprocessor. That's this first step. And then we're going to talk a bit about formation of the so-called RNAi-induced silencing complex, or the RISC. That is the process by which the dice small RNAs get into the organoid protein. And that's the limiting step to be able to do silencing. You can do all the cutting you want here. If that RNA doesn't get into organoid, no silencing is going to happen. Okay. And I mentioned this, lots of other non-canonical pathways. Now, I'm not going to talk about microRNA function per se, but it's dear to my heart. These are some recent works we have on microRNAs do interesting things in biological setting X, Y, and Z. And something to keep in mind for the second half of the talk is that flies have a distinct RNAi pathway. It's one of the cases where invertebrates are actually more complicated than mammals. And all would know that with SMART pathways, C. elegans has bizarrely complex SMART pathways, more so than in humans and mammals. People think, oh, invertebrates are very simple, but actually in small RNAs, most invertebrates are much more complicated than what occurs in mammals. So, we started with microprocessor for the reason that some years ago it was recognized that there is hemin inside of microprocessor. This is an oxidized heme molecule that is bound by the cofactor of drosha called DGCR8. DGCR8 is there's 2 copies of that and 1 copy of the RNase3 enzyme. And it was shown by a number of different laboratories that what hemin does is to probably stabilize this complex, allowing this cutting event to occur more efficiently. And the second thing is that it also positions microprocessor to cut accurately. The target genes that you regulate are set by the register of the first nucleotide of the microRNA. So if you don't cut that first nucleotide to the T, you're going to regulate the wrong targets. So you have to cut accurately and you have to cut efficiently. So this is definitely the case, been studied by many labs. What I realized was that as a geneticist, nobody had tested, do you need heme synthesis per se, genetically, for microRNA biogenesis? It's all done in vitro with purified proteins, and you can purify these recombinantly with and without hemin. So these properties are definitely true, but no one seemed to have tested, do you need heme synthesis in cells for this? It seems like, what's the answer going to be? Well, of course you're going to need it, but actually it turns out sometimes when you test something which hasn't been addressed, you're going to find something new. So I didn't know much about heme synthesis. It turns out it's very complicated. I don't know why it's so complicated. It starts off with glycine and succinyl-CoA inside of mitochondria, and then there's a chain of 8 enzymes that take the next enzyme product and then does something else to it, and it moves outside of mitochondria and then moves back inside of mitochondria. Finally, you get heme. Okay. Each of these enzymes is essential intracellularly to make heme. You can't bypass any of these steps. It's a linear epistatic pathway ending up with this heme molecule. However, in some sense, the cell is treating the first enzyme, ALAS, as the most important step of this pathway because it is using ALAS enzyme level to set heme synthesis. So it's rate limiting, meaning if you make more ALS protein, you're going to make more heme. It's also feedback regulated. So if the cells recognize there's too much heme, it turns down this enzyme, and then you make less heme. So it seems like cells go into a lot of trouble to set how much heme you're going to make based on the level of ALS protein. Everything else, to some extent, is less regulated. So Sanjay Lee in the lab made HEC cells that do not express detectable ALS1 protein. And then he tested microRNA levels. And again, microprocessor contains heme. It's the first step in microRNA biogenesis. So the expectation was you would accumulate the primary microRNA transcript that it normally liberates the hairpin from. And using these knockout cells, Sanjay sort of surprisingly found there was no effect on a number of primary microRNA transcripts by qPCR in these mutant cells. And he tested this a little bit more carefully. He put a microRNA into the 3' UTR luciferase molecule, and if that microRNA gets cut out, it won't make the luciferase. So we can monitor luciferase, and still no change in this microprocessor sensor. And then he knocked out another member of the pathway, CPOX, essential to make heme. That has no change in the sensor. And at this point, it was very, very curious why there's no change in the step that's supposed to be heme regulated when we have these knockout cells. The other part why I quiz people as to, do they know the problem with this experiment as set up? Because like, it's very sad, we spent a lot of time thinking about this, not realizing that we had shot ourselves in the foot with the way we were doing this experiment. Does anybody know what the problem is that we were trying to test? What happens to microRNA biogenesis when we knock out essential heme biosynthesis factor? We have the validated cells and none of this protein detectable. Compensation? Compensation. Where could the compensation be coming from?
Yes.
FBS, it turns out, has a huge amount of heme in it because it's bovine serum. It's got all the exploded blood cells, red cells in there. There's a lot of heme floating around in there. So we didn't really quite realize this for a while, that there is some orders of magnitude more free heme in FBS than the cell can even tolerate. So it's just taking up heme from the outside. So you have to get rid of this heme from the FBS. And if you do heme depleted FBS and then you grow the cells in, you get kind of the expected result. The primary microRNA transcript accumulates 50% or so. So that was, okay, so now we're like a year and a half into the project. Now we've recapitulated what was supposed to happen, which was nice. But then we had all these cells and we were wondering, is there a defect in microRNA biogenesis under regular cell culture conditions where it doesn't look like anything is happening to the heme regulated step? This is where everything started turning topsy-turvy and we started to get like the completely opposite results of what we expected, which I love because it means something new is happening. So heme was supposed to be a positive component to make microRNAs. When we knock out intracellular cord heme factor, we should get less microRNA, we actually get more microRNA. So this is with northern blotting of total small RNAs. This is with microRNAs that are inside of the effector AGO2, there's more, and it's also true in genome-wide studies. We see a directional shift that most microRNAs are accumulating. And I'm not showing it here, but we've also measured the occupancy of AGO2 on the transcriptome using a method called variation of clip, and there's more AGO2 on its targets in these cells that are knockout for ALS1. So somehow ALS1 has an unexpected role to restrain the microRNA pathway. And we don't know how it works, but however it works, it has to be opposite to the expected role. So we assumed it was not here. We did some more tests biochemically. These cells do not have a defect here. They don't have a defect here, but what we found is there's a defect in this risk loading step, getting the small RNAs into Argonaut. This works better when you don't have ALS1. This is how we measure this. We can assemble risk in vitro using radio-labeled duplex, and then incubate that with lysate, and then we're measuring the formation of the single-stranded form of the Argonaut risk complex. You need to be single-stranded to be able to base pair with targets, and that runs faster on a native gel here because it's lost one of the strands. So if we do this assay from lysates that are knockout for ALS1, we get about twofold increase, which is not bad because the other aspects of mechanisms we tested, there wasn't any change. And we're looking for something that's increasing microRNA biogenesis activity. That's a very critical colleagues, and they said, is that all you have? Just twofold. You tell me that's going to explain everything. And for anyone who's done this kind of an assay, it is known to be very inefficient. This is the free siRNA that we're putting into this. So most of the siRNA is not being incorporated in complex. And we now recognize very well why that's the case, because in mammalian cells, almost all of the Argonaut already has a microRNA in it. And it's very hard to get the microRNA out of it. So when you do this assay, you're working with the lysate of the cell, but a very tiny portion, maybe a couple of percent, is what my colleagues say, is potentially the free amount of Argonaut in a mammalian cell. That's what you're trying to load into. So we try to sensitize this a little bit by decreasing flux into the pathway by doing a transient knockout of core microRNA factors. So in this case, we did Drosha because we're working with Drosha for other reasons. It says guide RNA CRISPR-mediated targeting is transient. There's a clear decrease in Drosha protein if we do the lysate from this situation, there's again about a two. This is what happens in ALS1 knockout. This is about twofold if you quantify it in replicates. So we do get more stuff into Argonaut, but not like a whopping amount. Because again, in the time course of this, quite a bit of the Argonaut protein still had a micron in it. But if we do this Drosha CRISPR on top of ALS1 knockout cells, look at this, bang, it's beautiful. So now we really see how much more risk you can form of the available risk in a cell when you knock out ALS1. And that seems to be very consistent with the profiling studies we have done. Okay, these are knockout cells that we've selected, stable knockout cells. Remember I told you that cells are monitoring the heme levels, and then they're setting the new set point for heme biogenesis by adjusting ALS1 protein. So you can also decrease ALS1 protein by dumping more heme onto cells. So if you put a lot of heme into the FBS, more than it already comes with, you see that cell turns down ALS1. So we're quite curious what happens in this situation, because I said, well, if it decreased ALS1, you get more microRNAs. So we checked out microRNAs in cells where we put an excessive amount of hemin onto it. It's easier to get these solution using hemin. And actually the same thing happens. So we get increased microRNAs in this heme overload situation. So by some genetic hierarchy, ALS1 produces heme. Heme also represses ALS1. And now I told you that when you put too much heme on, you also seem to enhance risk formation. So are these two arrows, or is this happening through ALS1 protein? And I realize we have a very convenient way to test this because we have knockout cells for ALS1. So we can test the epistasis of this. So if we do this on top of ALS1 knockout cells, there is no added effect of heme, increasing heme, if you don't have ALS1. You just have the same result as ALS1 knockout cells. So we believe this is evidence that actually, although ALS1 initiates heme biogenesis, it is downstream of heme for microRNA regulation and suppression. That might suggest that this has actually nothing to do with heme. These are different ways to modulate the level of ALS1 protein. And I'll just show you one test that seems to be the case. We've done quite a few tests. Remember, we made these CPOX knockout cells. Where's CPOX? Over here. Okay. When you knock out CPOX, the cell is sort of thinking, I don't have enough heme, and so it turns up ALS1, right? So now we have this huge amount of ALS1 protein. In the end, you cannot make heme intracellular. You're going to be blocked at various intermediate steps, but you don't get to the end of this pathway. So we tested single and double knockouts for all the microRNA assays I just mentioned. And only when you knock out ALS1, either together by itself or together with CPOX, do you get increases in microRNA levels and in microRNA activity, but you do not get this effect when you knock out CPOX by itself. A variety of other tests indicate that ALS1 is the only member of the heme biosynthesis pathway that exerts a negative regulation of the microRNA pathway. So we're very excited because it seems to be the first moonlighting function of ALS1, previously known only to kickstart heme biosynthesis. Okay, this is all in cells. I mentioned as a geneticist, I'm always curious about more in vivo settings. And so we endeavored to see, does this work outside of cell culture? And here's where being in New York City turned out to be a great place for this, because just up the street from us at Mount Sinai School of Medicine, as I did some Google searching and PubMed, I found the world's experts are just like 2 miles away from us. The world's experts who studied this gene, ALS1, in mice and humans. And we were able to enter a wonderful collaboration with Bob Desnick, who's really founding member in heme-related diseases in humans, the porphyrias. His former postdoc, Mikiko Yasuda, has her own lab now, and Mikiko's postdoc, Sengi, they've been working on a conditional knockout mouse for ALS1. So if you're a straight germline knockout, you die quite early in development. But using a conditional knockout, they can remove it tissue specifically. and it wasn't even published yet, but they were very kind and open to doing a collaborative experiment. So they happen to be working in hepatocytes. They use a Cree called serum albumin Cree to flux out this ALS1 in hepatocytes. Now, I have to mention one thing that you may have been wondering. There's a 1 after ALS. There isn't a number after the rest of the heme biosynthesis members. And that is because there is an ALS2. Does anyone know where ALS2 is expressed? Maybe some of the
Red blood cells?
in red blood cells, right? Because you have to make this huge amount of hemoglobin. So ALS2 is just like another crank to up this pathway so you can support heme synthesis in those cells. Now, it was thought that that's the place where ALS2 is expressed very specifically in erythroid cells. But it turns out there is some in hepatocytes where they happen to be studying. Okay, so there's a paralog ALS2. For most, it's not expressed in HEX cells, so there's only ALS1. We knock it out, that's it. But in hepatocytes, there's another basically identical enzyme. So to do the experiment, they have to knock out ALS1 with tamoxifen-induced Cree expression, and then they have to come in and knock down ALS2 with an siRNA. And then we want to look at their hepatocytes. So here's some validation that this works. The ALS1 protein is gone in the conditional knockouts of purified hepatocytes. ALS2 is gone when you hit it with the siRNA to ALS2. So now we can see what happens in these livers. They take the livers, they isolate hepatocytes, what's going on with microRNAs in the ALS1-2 double mutant. And amazingly, same thing as what I showed you in tissue culture, was that microRNAs go up. Each of these is an independent liver from a different mouse. So you can see all the double mutants have increased levels of microRNAs. In sequencing, again, there's a directional shift towards upregulation of more microRNAs. So that's amazing. Same result in vivo. The last little bit for this section of the talk is now we have this interesting knowledge on this break on risk maturation and microRNA function. Can we harness this somehow in a therapeutic manner? Now, for those who followed the therapeutics of small RNAs, actually I have to lecture on this to the graduate students. I just did my lecture last week. For the first 13 or so years, I would give the lecture, I would end it by saying, there's a lot of promise people have been thinking about pretty much from day one. Can you use RNAi to knock down some genes that knock down oncogenes or some metabolic genes that have some kind of disorder associated? It just never was happening. We could deliver things very well in tissue culture or in worms or in flies, but like getting it in humans is quite a different question. So there's several challenges. siRNAs by themselves are not bioavailable as you get degraded, so you have to protect them somehow. And then you have to get them into the cell type of interest, the targeting. And then eventually they have to be functional. They have to get into a RISC complex to mediate silencing. So it turns out that these two have been worked on a lot. And because there are solutions for these now starting in 2018 and then annually thereafter, there has been a train of FDA-approved siRNA drugs that mostly treat liver diseases. There's a lot of stuff filters to the liver, so it's very easy to get stuff into hepatocytes. So there's a variety of siRNAs, clinical siRNAs, that are delivered in various methods to treat these kinds of metabolic disorders. And amazingly, it turns out that the second one of these targets this gene I've been telling you about, ALS1, which is a core member of the heme biosynthesis pathway. You might say, does that sound like a kind of insane thing to target an essential gene? But actually, that is the first-in-kind design drug that treats this condition where you have too much ALS1 and it's associated with porphyria, it's an imbalance in these porphyrin molecules. So it is clinically, every drug in principle should have some side effects, but that makes patients better. It's like many hundreds of patients. That's the only thing that is a treatment for that. So, we are wondering, are there any effects on microRNAs when people have been treated with that? And I've been told there's no test of that. In fact, there's no test of microRNAs in any of these conditions in humans. have to biopsy a piece of liver, like no one wants to do that. So they're just testing, are you okay? And if you're okay, drug moves forward. But it actually is quite interesting, given that, generally speaking, in control patients, it's okay to have this drug. And now we're wondering, could depletion of this actually enhance other siRNA therapies? Because there are 5 drugs, but there's not 500 of them. And one of the reasons is you can't effectively knock down the genes in context. So anything, I think, would help. So this is the intracellular boosting of RNAi hasn't really been addressed, and we did a few experiments to kind of see if we could support that. So in knockout cells, if we express a microRNA and give it an artificial target, we see more silencing from an introduced microRNA. And in mice, in this condition, where we have this conditional knockout of LS1 and then knockdown of LS2, if we challenge with another siRNA, indeed, we can get enhanced silencing. Although this is a little bit of a jiggered situation because we have to make this siRNA at a lower amount so that we don't have totally efficient silencing. But in fact, it is the case. If you have a suboptimal siRNA, it will work better if you don't have LS1 and 2. Now, it's been pointed out to me that making floxed humans just so we can use this is not a very viable approach, but there is a drug which is an siRNA LS1, and actually it's the same sequence in humans and mice. So those studies were done in mice. If we deplete with Givosiran, the human drug, and then the siRNA against LS2, and then come in with the third siRNA, what's going to happen? And for those of you who thought about microRNAs, the traditional dogma is that there's a zero-sum game. If you put in a lot of exogenous stuff, you're going to be displacing some normal endogenous microRNAs. But here we're hoping that with evidence that actually make more risk, we might be able to enhance RNA. So this is a triple siRNA situation. In fact, we still get this enhancing effect. It's very surprising. I think it supports our idea that actually we're able to make more risk in this situation. So yeah, so that's this point I'm making, that there might be sort of a hidden benefit of using this is you can boost the amount of risk that's formed. Okay, so for this first half, I told you how we found an unexpected suppressor of microRNA function. It's this initiating enzyme in heme biosynthesis that somehow restricts the efficiency of risk formation. And interestingly, it seems to be a non-canonical role of this that's unconnected from heme synthesis. And heme does have an effect, but only because we're changing the amount of this protein. And we're trying to exploit this to enhance therapeutic silencing. So we're working more on the molecular mechanism of this and trying to get some support so that we can do some more expensive experiments that have to do with looking at this as a clinical benefit. Because the next part is totally different, if there are any questions, I can take a few questions now.
Great job. Have you tested thrush-independent microRNAs?
Thrush-independent or?
Thrush-independent microRNAs.
Independent. Yeah, we tested a number of different triggers. That one is actually a very tricky trigger, which is independent of drosha and independent of dicer. We developed this kind of a vector some years ago. So it works with canonical microRNAs and also some kinds of non-canonical microRNAs.
This is the same effect.
Yeah. It works also with duplexes, so like you don't need to make it.
And LS1 is in the mitochondria.
Right. We tried in the mitochondria. I don't show some of this data. Its only known role is to initiate heme biosynthesis inside the mitochondria. So it's a nuclear gene, so it has to be made in the cytoplasm and eventually get imported. We have some evidence, very curiously, that if we cut off the mitochondrial localization signal, it can still support this role in inhibiting microRNA function. So we think it actually would be the first role a functional role that is not in the mitochondria. We haven't totally nailed that, but we have some very intriguing evidence that it's a non-mitochondrial role for this enzyme.
You probably have some protein-protein interaction. Does it interact with ADO or?
Yes, that's very sad because it's not like a year testing every single known thing, we cannot get a stable interaction with anything in the microRNA pathway. So that's there's a question mark exactly what is happening. So it's either very transient or there's another intermediary factor that's doing the job and we haven't identified.
Very nice talk. You showed that when you did with LS1, you have more microRNAs. Did you check what happens when you overexpress LS1? And for example, in your another mutant specs, you see more LS1 expressed. Do you see depletion of microRNAs as well?
Yeah, so how much of a rheostat is it for this? Can we sort of get more or less? There's a technical issue, which again relates to, I keep saying that microRNAs are very stable inside an organoid. So when we make more, we can detect that. It's hard to see less because you already have the microRNAs in the cell. And if you express ALS1, it's very toxic to cells. Okay, so when you take away Cbox, it causes phenotypes, but not because of the heme problem. Because the ALS1 its downstream byproducts are toxic to cells. So there's a lot of kind of problems with trying to do that to make the cells live long enough to turn over them. There are ways we could monitor nascent microRNAs. We haven't done that. So there might be a little bit of effect, but it's not like a huge effect. But there's technical issues to be able to quantify that the way you want to.
I have a related question. So heme is very important for mitochondrial function, right, and respiration processes. So
there are many dozens of heme proteins.
Yeah. So how are these side effects not taking over? Is it really just dosing? You're doing it at a time point and for a short duration enough?
Yeah, we have data. I hadn't shown it, but when you grow them under regular FPS conditions, there's plenty of heme. So we quantify heme. Under those situations, intracellular heme is fine. Chucked a bunch of hemoproteins, they're all fine. So it's really the protein itself. It's not its flux of the heme pathway, because they get the heme from the medium. Yeah, so we've tested a number of things. There isn't any change in intracellular heme, as long as you can take it up from your environment.
So you think this mechanism is conserved in flies and other invertebrates?
Yeah, that's an ongoing question. I had a little conversation this morning. So in worms, unbeknownst to me, until you start working on something I hadn't worked on, it turns out worms ditch the entire heme pathway. They have to eat the heme and then transport it through their gut. So it's definitely not working in worms because they don't have any that lost all of the enzymes. It's amazing. And other species are under investigation, but we have human and mouse so far. Okay, yeah, the last point. So it wouldn't be the first time that there's sort of interesting side effect of a known drug, so we'll see where this goes. Second part now. We want to look at the biology of mRNA-mediated sound sync. And this is not microRNAs, this is really the RNAi pathway. And as I said, we kind of got into this, started with the biochemistry, and then we're looking for a phenotype. And this is what the phenotypes told us. That's why genetics are so important. It's like a word to reveal RNAi and microRNA beam regulation. So the context that we come to is sex chromosome conflicts. You know, there's a lot of sort of reasons we can think that the X and Y have sort of different interests, if you will. They're very different for gene content. That imposes very different chromatin structures on the sex chromosomes, on the X and Y, and very, very different evolutionary trajectories. The Y, after it's been born, evolutionary is sort of on a, after it starts to lose its genes, keeps some male determinants, sort of on a road to death. And then a new sex chromosome will have to be born. And so this is a very fast evolving chromosome. And these settings can be hijacked by genes. And it leads to this idea that there could be selfish genes which are mostly trying to propagate themselves, but are actually not contributing to the fitness of the organism. In fact, they could be detrimental wild-type functions. I think of selfish genes, most of you might think of transposons, Nelson, or viruses, because we've all had COVID 8 times, and the flu 30 times, and constantly being invaded by selfish genetic material. But what about the enemy inside? Could be genes in your genome basically trying to hijack you to spread themselves. This Dawkins selfish gene hypothesis. So there will always be cheaters. We learn that in life and also in genetics. A gene can hijack sex chromosomes by living on an X chromosome and then figuring out how to toy around with inheritance such that Y-bearing sperm are disadvantaged. If a gene can innovate that activity, it's going to be evolutionarily very powerful, because when it passes through a dad will have inability to pass on the Y chromosome, all the gametes will be X's, and now we have all these daughters. So that gene is just spread unfairly throughout the population. So something that can do that can really spread through the population very, very quickly. That's good for that gene, but obviously it doesn't seem like it's good for the population to keep some of that around. So that time point places a huge pressure to find a suppressor before he went extinct. And if you're able to get a suppressor and kill that gene, then you would get back to this situation. And now it looks like nothing is going on. And so it's actually very hard to realize that this kind of conflict is happening, because if it becomes too powerful, it went extinct, and if it's under control, it's in a cryptic state, and you don't see the phenotype unless you really, really were looking for it. And you might think that they might be rare if you never heard of this. Actually, they're very broad across all domains of sexually reproducing life. It's just that most of them, the loci were never cloned, and we don't know what they do, and they more or less live in cryptic situations. But they occur in plants, and vertebrates, and mammals, and fungi. So I like to pay homage to Yun Tao because he identified loci that mediate this in Drosophila, but not in Melanogaster. Melanogaster doesn't seem to have the system, but in the species that is the closest sibling to Melanogaster, called Simulants, he found evidence for a selfish gene on the X chromosome. He called the distorted on the X, OX. It suppresses Y-bearing gamete formation, and that is suppressed by an autosomal locus called not much yang, or NIMI. And amazing, when he mapped these loci, he found that there's complementarity between the suppressor and the driver. Here is the suppressor here. It has the possibility for an inverted repeat structure, which contains homologous sequences to the DOX gene. Now, at the time, it was not known, is this involving a small RNA pathway, but certainly we know others have thought about that, because that's how microRNAs and RNAi works. These are the pathways in flies, and the microRNA pathway, if you delete any of these individual components, generally animals are lethal, and you know from Nelson's work that many RNA factors are sterile in one or sometimes both sexes because of transposon derepression. But the main phenotype of RNAi deficient flies is that that's a bad background to do your knockdown experiment. It's not going to work, right? You can't do experimental knockdown, but the animals themselves are kind of okay. So much so that people wondered, are there actually endogenous sRNAs? Where you're just waiting to be hit by a virus, for example. Then you're going to have a problem. You need RNAi to defend against viruses. What we noticed is that there's a class of sRNA substrates that we discovered some time ago that we called hairpin RNAs, and the NIMI locus in simulants looks like one of these hairpin RNAs. Now, this is some data that shows if you knock out that one hairpin RNA called NIMI, which we now classified as a NIMI, the males are fertile, but they give rise to almost all daughters. This is the sex ratio bias that a dad can only have daughters. This is posited to be a release of the suppression system so that some bad gene becomes active. Now, we investigated NIMI. We showed that it's a spliced mRNA that has an inverted repeat on it, and those are where the small RNAs are made from, and the different mutants produce the small RNAs, and that these are actually not microRNAs. We can distinguish microRNAs and sRNAs because they have a different pathway. They go into a different effector called Argonaute-2, and they receive a 3' modification that only sRNAs and RNAs receive. So it's definitely an sRNA locus, and it can directly silence the DOX gene. With our hands on the NIMI hairpin RNA sequence, we're able to find another hairpin RNA, which is only in simulants, not in melanogaster, and they clearly have an evolutionary relationship. And they make different kinds of sRNAs, even though they have sequence homology, and they sit in a very interesting region that had been mapped also by Yuntao as housing some locus that also suppresses XY conflict. So over the years, we have knocked out both of these herpin RNAs in a clean genetic system, studied what they do, and the end picture is that Drosophila similans looks like it's a perfectly happy fly species. It's pretty recently born. It's thought to have been born on the islands off of southern Africa about 250,000 years ago. And since then, it has spread everywhere where people are. It's a human commensal. It's gone around the planet. So it seems like it's great. It can colonize all sorts of new territories, live on stuff that people eat. But we think it's just hanging on by a thread. And that species is like on the brink of going bye-bye because there are a bunch of distorters on its X chromosome, and they have to be suppressed by de novo sRNA loci to keep them silent so that you never knew they existed. We can reveal their function in knockouts of the Nemi herpin RNA and the Timi herpin RNA. Yun's genetic work showed that if you remove Nemi, dad can only have daughters. More recently, we knocked out the Timi herpin RNA, and that's more perplexing because it's viable, no phenotype in females, but in males, it's completely sterile. You can't have any kids. And it's a real disaster in spermatogenesis. There are no sperm. These little blue needles in the end are sperm in the seminal vesicle, and they're immature sperm in the Timi knockout testis. So envision that if we take away this hairpin, all the gametes are destroyed. Remember, I'm positing that this is a protection against a meiotic drive gene, but that gene just suicided its own transmission. Does that make sense at all? And I think there's actually a very good analogy to that recently, because if we did not develop mRNA vaccines or herd immunity to COVID-19, it could have wiped out the humans in 2020, right? And then when all the hosts are gone, where's SARS-CoV-2 going to do? Maybe it'll jump to another animal, but like, it's very possible for a selfish gene to suicide the population. It doesn't really know what the end outcome is going to be. It's just trying to spread. Now we have some good evidence that actually this is still trying to suppress a sex chromosome conflict. And that comes from a genetic interaction experiment. I didn't tell you that this knockout is temperature sensitive. At 17 degrees, it's 95% girls from the dad, but you get a measurable amount of sons as you raise the temperature. This is actually a normal culture condition. It's not heat shock. This is a normal temperature. And somehow that makes the system temperature sensitive. So what we do is remove one copy of the Timi locus, heterozygote. By itself, that has no effect that we can tell on progeny. But if we make this heterozygote in the Nemi knockout, under all conditions, you're now back to almost all females. So we think that basically it sounds a X distorter, but in the lab condition, we sort of unleashed this monster, and now it just totally ablated gametogenesis. The other analogy I make for this is these ones here. Think of the dog's family as running after the roadrunner. That's the story. He's trying to kill this, right? But we know what happens is it comes with all these ridiculous contraptions that keep blowing up in this space. So we think that under the lab situation, it's trying to chase after and kill the roadrunner, but it just suicided everything. That's the analogy. Okay. Because we have this full sterility, we imply that it should target something else besides the DOX gene. It turns out there's another paralog of DOX found by Yuntao called the mother of DOX, or the NDOX gene. But when Jeffrey Veneniagin was in the lab, he hunted around the newly assembled simulant's genome further, and he found that there are 2 more copies on the X of genes that are in the DOX family, but they have complementarity only to the Timi herpin RNA. They seem to be selective targets. We call these the pair of DOX genes. All these DOX genes live on the X chromosome, so they're fitting the idea that X is making these different genes to fight against the Y chromosome. And it turns out they're also highly polymorphic. These are natural deletions in different wild lines of the DOX or NDOX or the paradox loci. And that enabled a genetic test with Colin Nickeljohn's lab, if we take an X chromosome, which is deleted for some of these, and that's the main thing that this needs to suppress, can you rescue the hairpin RNA knockout by removing these genes? And we have some very interesting data that, in fact, if you use the right X chromosome from a wild, like collected from a wild animal, that's deleted for some of these, you can rescue the Nemi phenotype. More surprisingly, there is an X chromosome which is deleted for DOX and for PDOX2, this genotype, that rescues Temi fertility, and these actually have equal sex ratio. They fix the sex ratio defects. So we think the very interesting X chromosome, it's basically broken from all the drive systems. So we're studying that further. Now, I didn't tell you what the DOX genes encode. In fact, it wasn't clear for some time when Yun Tao first identified them. He thought it could be that they're non-coding. Staring at these genes for many years and found more copies of them, found out there's actually quite an identifiable domain, which is very curious because it's a DNA binding domain, something that could affect chromatin. And its closest relative is something called protamine. And we know that these things are related to this HMG box domains. Playing with AlphaFold, even though those are de novo gene products, they fold right onto the AlphaFold structure for HMG box domain proteins. These are in humans. But they came most recently from a gene called protamine. And protamine is something that repackages the male paternal genome. All the histones got taken off, and then they get hypercondensed using protamines so they can fit into the sperm head. And it seems to be about the time, the so-called histone to protamine transition, when the DOX proteins are expressed. So we're very curious. We don't have functional data at this point, but we think that basically they're hijacking something about chromatin packaging in dad's genome to do something bad with gamete formation. The last bit of data I'll show you is that we think these are the right genes to go after because they are running like madness, like craziness. So this is a section of the X chromosome from Lanogaster. All these genes in blue are conserved in most fruit flies. Simulans is the closest relative to Lanogaster, and it made a gene here, made one here, made one here, made one here. Now, there are two other relatives of Simulans called Mauritiana and Seychellia. And if you look at their regions of X chromosome, they've invented many, many more of these, and they're interspersed. If I didn't show you what this was, you might think, oh, these are transposons. They're not transposons. They're like protein coding genes. Okay? So they're amplifying within species. And that made us wonder what's happening in individual wild species, because I mentioned we found that wild X chromosomes can really modify this phenotype. And Grimasectia with long-read genomes that are made by Bernard Kim found that it is a complete mess. All these are wild Simulans strains. We found at least 7 more of these in different wild individuals, and more copies of these. So one here, here, this one down here. So there's at least 7 new ones. This fits what we call a Red Queen's arms race, that basically the X is inventing all these weapons to kill the Y, and then you have to bring that back to a steady state by killing all these genes using RNAi. This is the madness of Mendel's laws that can be hijacked. Okay, so to summarize this part, I've introduced you to a very bizarre class of genes that can mess around with meiotic transmission. They look analogous to virus and transposons, but they're really part of the self-genome. A set of these were born very recently from a chromatin packaging factor called protamine, and they're amplifying within genomes even though they're not transposases. So we don't really quite know how they're amplifying, but they're definitely amplifying a genomic copy. And they have interesting but unknown activities to kill Y sperm, which we'd love to recruit somebody to figure this out. It's very fascinating. Nobody's studying right now. The last parting thought is we think that this combination of the inevitable churn of the X chromosome trying to hijack gametogenesis for its own benefits, and then you need to silence that, could be an intrinsic engine, a molecular engine for speciation. Because I already showed you that in the right combinations, you can create sterile individuals, and you can also ones that have an antidote and are fertile. Right? So that's already been broken apart the species by the activity of these what nominally were sex chromosome drivers. So we're looking about how to generalize this kind of species. Okay. to summarize these two parts, we like to use a phylogenetically broad approach in invertebrates and vertebrates to learn about RNA microRNA regulation. We have this inhibitor of the microRNA pathway in many mammals, we know about invertebrates, with an interesting possibility to enhance RNA therapies. And then totally de novo, just evolutionary, just like yesterday these genes were born, they can cause all kind of havoc in the male germline, and these are silenced by endogenous RNAi lysine. First part of the talk was done entirely by Sung-Jae Lee, who's on the job market. This dude has 5 first author papers. Somebody should hire him if he doesn't get a job. It's going to be a mistake of the system. We had a wonderful collaboration with Bob Desnick, Mikiko Yasuda, and Mikiko's postdoc Sang-Mi. Second part of the talk, most of the bioinformatic work was done by Jeffrey Vinayagam, who just started his lab last year at University of Texas San Antonio. We have many friends that help us. We call it Nickel John's lab, Patricia and Holly doing some of the interspecific crosses. Bernard Kim has sequenced many genomes for us, and Marion working with Raphael and Benjamin, and have done some testis stainings that I can show you. And, thanks to the rest of the lab. If there's any time, I know I'm a little bit over, but I'll take a couple extra questions.
The second part.
Do you think there's any virus resistance that these meiotic drives might participate in? Are there viruses that infiltrate the simulants ovary like they have in Drosophila that maybe these things are also targeting?
We don't have any evidence. No evidence of that so far. I hadn't thought about it, but as I'm just thinking about it right now, actually what these genes do is they drive more females, and actually that's what some bacteria like to do, and probably other horizontally transmitted things. So Gypsy is kind of an endogenous retrovirus. They want to favor female production because they transmit to the next generation. The dad doesn't transmit these, so it's almost like it would help. So I don't know, maybe there's some way that these things get together that they help each other. But we haven't investigated.
Fascinating. Thank you very much.
Yeah, that's why we're in sperm. Fortunately, I can see data.
Oh, no, you will.
Okay, 2 days. That's good. All right.
So I think yours is the last
We're
of
going to lunch.
8 in the last 4 weeks.
Productive.
Well, let's see. Everyone having a
Yeah, so